Hi, everyone. My name is Jeremy. I work here on the communications team at the Molecular Foundry, and I'm here today for a conversation with Corie Ralston, who is the director of our bio facility, who does some amazing work with figuring out the structures of proteins. We're going to talk just about some of what she and the rest of the bio facility does, especially in the context of sustainability. So, Corie, can I have you introduce yourself?
Hi, my name is Corie Ralston. So I'm leading the biological nanostructures facility at the Molecular Foundry. I have a background in protein structure, protein characterization methods, and I'm thrilled to be here talking to you today.
Awesome. Wait, so can you explain a little more what proteins are and why it's important to understand them and just know about their structures in general?
Yeah, sure. So proteins exist in all animals and all plants and all bacteria and all archaea, and they're responsible for making life happen, basically. So in plants, they make photosynthesis possible. In the human body, they make it possible to ferry oxygen around from your lungs to the other parts of your body.
So basically just the small machines that keep everything running that is alive.
They are the small machines that keep everything alive. That's a great description for proteins.
Okay. Excellent. So let's see. So what does their structure have to do with their function?
Yeah, so it's their structure that really determines their function. So all proteins have a unique structure. So the structure is what basically tells us how the protein functions, and it tells us how to control the protein, how to inhibit it or activate it somehow.
Got it. So given that proteins are kind of they're the functional unit, all these different processes, both good and bad in our body, you're saying we mostly need to know their structures so that we can figure out both what they do and then what they interact with, and how we can purposely interact with them by, maybe making something that attaches to that structure or complements it in some way.
Yes, exactly.
So I grew up in seeing in textbooks like all of these diagrams of protein structures, and I think it's safe to say most people have seen some depiction of that. But how have we arrived at that knowledge? Like, how have we been getting these structures this whole time? Is it something you can see under a microscope? Is it something you can see in action in some way, or how does it work?
Okay, so what scientists do to get a protein structure using the method of crystallography is they grow up E. coli, and they get E. coli to produce the protein that they want in a very large quantity, and then they break open those cells and they purify out that protein, and then they put the protein in a environment such that it's in a drop that's dehydrating over time, and as the drop dehydrates, the protein molecules move closer and closer to each other in solution. And if the scientist is lucky, that protein falls out of solution as a nicely ordered matrix, a nicely ordered protein crystal.
Wow. And then, okay, so the process, up until now, or continuing through now, involves basically getting bacteria to produce, supernatural quantities of whatever protein you're after by giving them the gene for it, taking that protein out of its living context, and basically kind of trying to crystallize it, much like you would have like salt crystallizing at the at the bottom of a glass or something.
Yeah, exactly.
So all of this, I guess, strikes me as extremely removed from the context in which proteins like normally operate and exist in nature. Is that a hurdle, or is that a challenge at all?
Yeah, I would say that's the challenge for the coming decades for scientists. We know a lot about protein structure for proteins in isolation, but really what we're coming into now is the era of trying to figure out how they interact in a real environment. How are they really interacting with other proteins in a cell? How are they forming complexes? How is their concentration affecting their function? So what we really need to do now is we need to develop methods where we can look at proteins in a more physiological environment, a more in vivo type of environment.
That is so cool. I feel like I never fully appreciated the fact that, all of these images that I grew up, seeing in textbooks, or kind of these descriptions of like, how things work within the cell, have kind of been cobbled together, backwards from really just looking at each of these individual pieces in this very kind of isolated, separate environment. It's kind of I guess amazing that that worked and that we know as much as we do, given how difficult it is, but also that there's so much more to figure out by looking at these things in context.
Yeah, given that we're really only looking at protein structure generally on a level like that, it's amazing what we've accomplished.
Okay, so with that as like the context, can you tell me a little bit more about X-ray footprinting and what you do at the Foundry?
Yeah, so X-ray footprinting is a method to figure out protein structure in the solution state. So whereas with protein crystallography, you need to get a protein in a crystal form, with this new method of X-ray footprinting, you have proteins in solution that you're looking at, and you're getting some structural information. So the way the method works is we take a protein solution and we irradiate with X-rays, and the X-rays will interact with the water in the solution, and they'll generate hydroxyl radicals, so an OH from a water. Okay. And the OH will modify the protein, but the key thing is that the OH only modifies the side chains of the protein where a water had been nearby. So when we use this method, we modify the protein with OH, and then we look at where those modifications occurred, and then based on where they've occurred, that tells us where the water molecules were around the protein. And that gives us structural information about the protein.
Cool. So you've got a protein in solution, you kind of irradiate the water around it, and the water kind of alters it by really just adding on to it. How does that ultimately help you figure out the structure rather than just giving you kind of a mangled water altered protein?
Yeah, that's a great question. So what we generally do with this method is we look at a protein in two different states. So for instance, if a protein is folding, it may be a long chain that can have OH additions along the entire length of the chain. But once the protein folds up into a distinct three-dimensional state structure, the folds of that protein will come together, and that will exclude water. And so we don't get any of those OH additions on the interior there. So we can look at where the modifications occurred, and we can figure out how the protein, how those three-dimensional structures were formed based on how the water is oriented at the beginning and the end of the folding process.
So, okay, so to use like a really crude kind of real world analogy, it's almost like taking a rope and tying it up into a big knot, and then like painting the outside of it, and then uncoiling the rope, and based on what is still painted, you can figure out what shape the knot was in to begin with.
That's a great analogy.
You're awesome. That's so cool. So what are some of the benefits of doing it this way as opposed to the old crystallography method?
Well, so one of the benefits is that the proteins are in the solution state, so they're free to move around. So we get a more accurate picture of their actual structure in solution, not in this sort of artificial crystal matrix. And the other advantage is that there's a possibility we can start doing footprinting on whole cells. So that would be looking at proteins in their actual native environment.
Wow. So you can actually do this theoretically, at least in vivo, like actually in a living organism?
Yes, theoretically we can. It's a very difficult experiment. So there's many years of work ahead of us, but that would be sort of my vision for the footprinting field, is to eventually be able to get protein structures of proteins in their totally natural environment.
Wow, that's fascinating. So understanding that there's a ton of cool work already going on with just understanding proteins in a new way, what are some of the things that are on the horizon or that you're excited about for the future, especially in the context of, I know the bio facility does a lot of work on sustainability and just kind of some of the next generation of materials or energy sources. Does your work and the kind of the world of protein structures have anything in the works for that?
Well, one, I think, very exciting area of research is looking at hybrid materials. So looking at structures that are made up of biological molecules and inorganic molecules.
Cool.
So one of the applications that we did a couple years ago is we looked at this bacteria which produces a protein that is able to shuttle electrons along its length. And what this bacteria does is it's able to live on rocks and minerals sources. And so what we did is we took the protein by itself in solution with some metals, and we were able to figure out how the protein interacts with that metal. And so that gave us information about how that bacteria is able to shuttle electrons from its rock source.
Wow. So these bacteria and using these proteins actually are able to use metals in a rock as an energy source?
Yeah, and it has some very exciting possible applications in developing microbes as energy sources.
That is fascinating. That's so cool. I never would have thought of that application of like just looking at, beings and life forms and like strategies that are so different from ourselves as a way to unlock just like a whole new category of abilities.
Yeah,
Wow.
exactly.
So how does that, I guess, what are some of the things in the works at the bio facility that kind of might usher us along on that path? Are there any tools in your toolbox that can kind of that can take advantage of what you're learning here?
Yeah, so at the bio facility, we have a number of tools for both designing and synthesizing biomolecules and also biological and inorganic hybrids. One of the major workflows on our floor is in designing and synthesizing these molecules called peptoids,
Okay.
which are very interesting polymers that have characteristics of proteins and they have characteristics of plastics.
So wait, so this is basically taking the idea that proteins themselves are, they're effectively polymers. They're long strings of something made up of a bunch of individual units, and trying to replicate that with plastics or, like a man-made molecule. Is that correct?
Exactly. Yeah, that's a good way to describe it.
That's
Yeah, and they're non-toxic, so they're not. They're stable and non-toxic.
Wow, that is fascinating. So what are some of the applications that we might see for peptoids or for, kind of understanding proteins better in the space of sustainability, or like figuring out ways to kind of combat climate change, or just figure out more sustainable materials, more sustainable workflows?
Yeah, so we at the bio facility are very interested in this whole area of designing biomaterials for specific applications. And biomaterials are complex materials that maybe mimic some materials you find in nature, but have been designed for specific purposes. So my vision of the future is that someone could come to the bio facility and say, hey, you know what? I need a membrane that's going to allow water through and allow chloride ions through, but not allow these other metals through. And I need it to be stable up to 120 degrees C and chemically resistant, and this particular size.
Yeah.
And it'd be like we would be able to say, oh, we can help you design that material. Here are the building blocks we'll use. We'll maybe use some peptoids for this part of it. We'll use some proteins for this part of it. We'll have some inorganic section of it. And we can predict that it's going to give you all those characteristics that you need.
Okay, so is really the goal of all of this understanding, all of this kind of coming to know how proteins function and how that's related to their structure, and simultaneously developing tools to replicate that structure and replicate that function, is the final potential there, or the end game, to be able to design things that mimic like the sustainability of natural materials with a whole new layer of customizability and kind of intention that we can program into it?
Right. So if we can design biomaterials where we know the components are basically regenerating or break down in a certain way, then we'll have a lot more control over what we put into the environment and the effect it has on the environment.
That's so cool. So I mean, you can use like the building blocks of just normal life to make things that are, formerly were the domain of much more kind of resource intensive, metals and machines. And
yeah, exactly.
That's so cool. Okay, well, this has been fascinating. I learned a ton about things I wasn't even expecting to learn about today. And I'm just so excited about the work you all are doing at the biological facility. And I really appreciate you taking the time to talk to me today.
Well, thank you so much, and my pleasure.